Good evening, everyone. Welcome to the Allen Institute. We are going to get started. So thank you, everyone, for coming out tonight for our public event, The Brain, IDK, what we know and don't know about brain cells. My name is Jack Granger. I am the Education and Engagement Intern here at the Allen Institute this summer. And creating this public event has been my project this entire summer, and I am so happy and grateful to see so many amazing faces here tonight. So thank you, thank you, thank you. Thank you. So before we get started, I just want to quickly go over an agenda for the evening. I'm going to start off with a quick welcome to give you guys a little bit of information about the Allen Institute itself. And then we are going to go straight into our first speaker, Dr. Rachel Hostetler, to tell us about what we don't know about brain cells. And after Rachel, we will have Jeremy talk to us about the ways that the Allen Institute is trying to answer what we don't know about brain cells. And then I will give a short conclusion where I tell you guys about some of the activities that we have set up for after the talks, and the food and drinks will also reopen. So yeah, those are our plans for the evening. So the Allen Institute was founded by Paul Allen, who is a co-founder of Microsoft, way back in 2003, when he gathered a group of some of the most prominent neuroscientists in the field to ask them what would further the field of neuroscience at this point. And what they answered with was, we need more foundational research that is open to the public. So the result of this was the creation of the Allen Institute for Brain Science way back in 2003. Over the past 20 years, the Allen Institute has grown exponentially. We are now at over 900 employees, and we are still growing. Part of that expansion means that not only do we study brain science, but we also study areas such as immunology and general cell science. During this entire time, we are a nonprofit, meaning we do not make money from the release of any of our research methods or resources. And now I'm going to talk a little bit about some of our core principles here at the Allen Institute. The first is big science. This basically means that the Allen Institute strives to answer some of the biggest questions in the scientific community, such as, what does every cell in the brain look like? Can we map an entire human immune system? In order to answer these big questions, the Allen Institute has to have teams of people with all kinds of different backgrounds. So this is where our next value comes in, team science. So as I said before, we have multiple different areas of research in the Allen Institute, and we actively encourage scientists from all different fields to collaborate on projects and to create new research. Our third value is open science, which basically just means that we like to share. As I said before, all of our resources, research methods, all of these things are free and open to the public. We believe that the best way to further scientific innovation is to make sure that all of our resources are open and available to researchers and the public alike. Our fourth value is impact. We don't do research here at Allen just for the sake of research. We choose to pursue projects that will have the biggest impact in areas such as health and education. So now I'm going to hand it over to our first speaker, Dr. Rachel Hostetler, who will tell us what we don't know about brain cells. Now you might see this QR code right here on the screen. If I could ask you guys to take it out and scan it with your phones. Throughout the talk, if any questions pop in your head, feel free to submit them on this form, and we will be reviewing them. And after Rachel's talk, we will have a very short Q&A session while I will ask her a couple questions from the form. So Rachel, take it away.
All right, thank you for that introduction, Jack. I'm Rachel, and my portion of the talk will be going over about what we don't know about brain cells, and then Dr. Jeremy Miller's portion of the talk will be how we're finding this information out at the Allen Institute. So first, before I go into what we don't know about the brain, I do want to mention we do know some things about the brain, right? What do we know? We know the typical size and shape. This is what a brain typically looks like. We know that an adult human brain is about 3 pounds. We know that it consists of a cerebrum, a cerebellum, and a brainstem. These are all different parts of the brain here. We know that within the cerebrum, we have 2 hemispheres, right? So the brain is divided into 2 parts. We have this big fissure down the middle. And we know it has these 4 different lobes as well, which are color coordinated here on this diagram. So this is what a typical brain looks like when it goes through all the developmental biological processes and develops to an adult brain. And then when this process doesn't go typical, there are some other things that can happen, right? We know if the brain is too small, That's a disorder called microcephaly. We know if the brain is too smooth, meaning it's missing all of those wrinkles and ridges, that's called lissencephaly. And we know that if a brain doesn't have two hemispheres, right, if it's merged together, that's a disorder called cyclopia. So these are all just some disorders that we know about what happens when the brain does not go through its typical development. What else do we know about the brain? We also know the general functions of different areas of the brain. So we know that the visual cortex processes visual information. The visual cortex is located in the occipital lobe, which is in the back of the brain. We also know that the brainstem regulates vital bodily functions such as breathing. We know that your cerebellum regulates balance and coordination. And this is important to know when there are certain injuries to the brain or diseases or disorders. So many people, when they suffer a stroke, the strokes often occur near the motor cortex, which causes motor and movement deficits. Frontotemporal dementia is a specific type of dementia that occurs in the frontal and temporal lobes of the brain, which specifically affects personality. So depending on where you have different injuries in the brain, you have very different symptoms. What else do we know about the brain? We know that the adult human brain has about 170 billion brain cells, and that about half of these are neurons, which are cells that send and receive electrical signals, and about half of these are glial cells, which are another type of brain cell that provides support to the neurons. And these estimates have varied widely over the last century. And why have they varied? Well, the technology that we have in 2024 is quite different than the technology that existed in the 1870s or the 1890s. And so as time goes on, as we've developed more and more techniques, more protocols, we've advanced our equipment, we're able to do experiments that are more and more accurate and get more and more accurate information. So in this way, scientific research is often self-correcting. So right now, our best estimate is about 170 billion brain cells. Now here's the part that we do not know. What are all these 170 billion brain cells doing? Are they all functioning completely independently of each other? Are they all acting together as one group? We don't quite know the answer to this. So this is the question we're trying to solve at the Allen Institute. What are all these different brain cells doing? And to give an example about these different brain cells, let's talk about another system we have in the body, the skeletal system. So in the adult human, we have 206 bones, and each of these bones have names, because we named them all. They have functions, and we know where they connect to, right? We know where the tibia and fibula are. We know the different parts of the skull, because scientists over the years have figured out how all the bones fit together. And this is exactly what we're trying to do with brain cells. We're trying to figure out of these 170 billion brain cells, what are their properties? What are they doing? What should we name them? We give them names, right? They don't inherently have names. Humans have to give names to them. What name should we give them that makes sense and that people agree on, right? And that's important because if you break your tibia, you go to the doctor and they say, yeah, it's your tibia. You know what you're talking about. This is a problem with brain cells. We don't all have the same names for different types of brain cells. And also, who do they connect to? So just like the skeleton, not every cell in your brain is connected to every other cell. They're organized in discrete circuits, and these circuits have different functions. So that's what we're trying to figure out, is how these brain cells connect and interact with other brain cells. And so we're working on identifying all these different parts of like the big brain puzzle. And so why don't we know any of this, why don't we know this information? Well, 170 billion brain cells is a lot of cells to characterize and to study. Also, brains are quite different than bones, right? Bones you can find thousands of years later. They're mostly, they're intact. You can look at them. Brains are a bit more fragile. They're more delicate. It's a bit more difficult to obtain brains and then to study them, right? The conditions you have to do it in research labs. Also, many scientists have really been debating the different names for brain cells for decades. So some neuroscientists came up with names in the 1800s. Some of those names we still use today in 2024. Some of those names we don't use anymore because we've updated them with newer names. So this is an active area of really debate, in the neuroscience field. What should we name this particular cell? And studying all these millions of cells together is actually quite difficult. A lot of the technology, equipment, protocols to do this type of research has only existed in the last 15 years or so. So with this new invention of new techniques, new machines, new analyses, we're able to gather more data. And then with these new data sets, we have another problem. Now we're dealing with petabytes of data. And a petabyte is 1,000,000 gigabytes. And so we have many, many petabytes of data in this building just on brain cells. And so that's an inherently different problem, figuring out how do we store this massive amount of data? How do we analyze it? What computers can run with this type of data? But at the Allen Institute, we're very specialized in tackling these big problems. So we have an entire team dedicated just to data and technology. We have another separate team dedicated just to histology, or looking at the brain tissue, right? We have these different teams that are highly specialized and focused on these different research areas. And they all work together, which allows us to answer these really big questions. And why should we care about this? Well, it's good to know what's going on in your brain, just in general. But it's also important in the context of disease. So there's been some very new research from the Allen Institute that's it was just accepted for publication within the last few weeks in a scientific journal on Alzheimer's disease that shows that not every brain cell is affected equally during the course of Alzheimer's disease. In this graph here, this graph is showing disease severity down here on the x-axis, and all these different colors here represent different types of cells in the brain. And from this graph, we can see that these orange cells here are severely affected in the beginning of Alzheimer's disease. These red cells here are more affected towards the end. All of these cells here on the left start to decrease with Alzheimer's, whereas these cells here on the right actually start to increase with Alzheimer's. These are glial cells, so these are different types of cells, and they actually go up in their number during Alzheimer's disease, right? So every cell is not affected the same in disease. This is important for developing therapeutics for different diseases, right? We can't just have one drug that targets everything. We need to look at what's happening on the cell-by-cell level. And so that's an example of why this research is so important. Why do we care about characterizing 170 billion brain cells? And so how do we categorize that? That seems like a pretty daunting task. Well, it turns out humans really like categorizing things. For instance, we've categorized animals, plants, types of bacteria, dirt. We're pretty good at organizing things just based on what we observe in the world. So humans were able to look around and notice some animals have a backbone, some don't have a backbone, some have legs, some don't have legs, some have 3 legs, some have 2 legs, all these different things. So just by observation, we're able to categorize all the animals. And this is the same thing that we're trying to do, but with cells in the brain. And so down here in this corner, we have us humans, which are in the mammal category. But for the next segment, I just want to focus briefly on another type of mammal, which is the whale. And I'm doing this because we're in Seattle and whales are awesome. So I just moved to Seattle last year, and I've seen whales 3 times in my life, and 2 of those times were in the past 2 months. So I'm a big whale fan now. So I brought them into my presentation. And while learning about whales, I learned that there's actually a debate going on about the orcas, specifically in Puget Sound, and that there's actually 2 different groups of orcas, right? And so a group of scientists submitted this scientific article a few months ago saying, we think these orcas are actually 2 different species, and we're going to argue that based on their morphology, so looking at their shape, the shapes of their fins, their size, the coloring, their behavior, so they eat different types of food, they have different behaviors when they're hunting, and also based on their genetic information. So they use these three aspects of the whales to say, these are actually different types of whales. And I'm bringing this up because we use very, very similar characteristics to look at brain cells. So for the whales, we're looking at their shape, their behavior, in their genes. When we categorize brain cells at the Institute, we are also looking at shape, behavior, and their genes. So neurons come in all different shapes and sizes, and we can do experiments here to actually detail and trace out individual neurons and measure all of their different little shapes, all of their different branch points. All these things are very unique to different types of neurons. We can also look at their behavior. So neurons don't speak to each other like the whales do or like we do, but they send electrical signals to each other, which is very similar to speaking. That's how they communicate with each other. So humans, we have different frequencies of our voices. Neurons have different firing patterns of their electrical activity, and that's what's shown here. We can see that this neuron here at the top in red has very fast electrical signals, whereas this one in blue has slower electrical signals. And we can actually identify neurons purely just based on these electrical signals. We're able to categorize them. And that's through a technique called electrophysiology. We can also look at the gene expression of our different neurons here. So what we have here on the right are two different plots, and these contain different information about millions of different cells. And the plot here on the top part here has red dots up in this upper part, which shows cells that are genetically similar to each other. And then down here at the bottom, we have this blue island, which shows that these cells are genetically very different from the red cells because they're in different parts of this plot. So just like the whales, looking at their shape, looking at how they behave, looking at their genes, this is really how we're looking at brain cells. And once we have enough information about all of our different brain cells, we can actually start to create a taxonomy. So similar to that animal kingdom taxonomy I showed you with invertebrates, and cold-blooded and warm-blooded, we can start to do that with information about brain cells. So here's an example of just that. This is looking at brain cells specifically in the motor cortex, so the area of the brain that's you use for movement. And this is all based on their genetic information. So in this taxonomy here, this chart, instead of having our vertebrates and our invertebrates, we have our neural cells and our non-neural cells. And then we branch down to our neurons and our non-neurons, or our glial cells, those support cells I mentioned earlier. And then we can keep branching down further and further based on genetic information. And with the experiments that we do here at the Institute, not only do we get their genetic information, we can also look at the shapes of the different neurons. So this figure here is color-coded so that any of the yellow neurons down here come from the genetic background of these neurons up here. And again, we also look at their behavior. We want to know the electrical activity of these different neurons, how they talk to each other. And we can see that, the cells in red have different behaviors than the cells in purple, which have different behaviors than the cells in blue. And by doing this in different areas of the brain and with more and more data, we can start to get a more complete picture of what the whole brain looks like. Okay, so that concludes my portion of the talk, but we have about 5 minutes or so for Q&A before I hand it off to Dr. Jeremy Miller.
All right, so we'll get started with some questions. The first one I have for you is, what is the most interesting fact about the brain that you think people should know?
Oh no, that's such a hard question for a neuroscientist. Man. I think there's a lot of interesting things, but one interesting thing is how specialized your brain really is. So in the auditory cortex, it's not just the area for hearing. You have specific areas that are responsive to certain frequencies of sound. It's that detailed, or different gradients of light. It's very, very specific in how it's laid out, and it's mapped out in this way where you have certain frequencies. Not only are they similar together in sound, but also physically in your brain. Hopefully that makes sense. But there's the way the sensory information comes into your brain is mapped out in a very distinct pattern. I think is super cool.
Yeah, I'd agree. The cochlea is definitely also one of my favorite brain structures. It's
yes.
fascinating.
Yes.
So my next question for you is, what are some common brain myths and can you debunk them? So for example, do we really only use 10% of our brain?
Oh, that would be not great if you only use 10%. Yeah, no, you definitely, use your brain. You don't just use 10% of your brain. I don't know, maybe you'd be in a coma or something. That would not be great. No, you use all of your brain. Your brain does different things at different times, right? Your brainstem's doing something completely different from your cerebellum, which is doing something very different from your thalamus, and you're definitely using all of your brain, just at different levels and at different activities, but everything's connected. So,
what about the left brain, right brain myth?
Yeah.
How do you feel about that one?
I mean, it's not so segmented. I mean, language is definitely on one side of the brain, but other things, like being more artistic or being mathematical, that's sort of up in the air about whether or not the left brain, right brain thing is actually true. And then I'm left-handed, so all those studies only usually look at right-handed people. So being a lefty, I'm like, well, does this even apply to me? So it's that's another question to ask.
For sure. Okay, this is my last question for you, Rachel. What advice do you have for young people who might want to be a scientist?
Oh my gosh, yeah, there's a lot of different ways to be a scientist, and you can have many different interests. So when I was in college, I majored in neuroscience and German, and I spent a semester abroad in Germany because I just really liked the language, and I got to work in a neuroscience lab in Germany, and then I got to do my PhD in neuroscience back here in the United States. So, but I think having multiple interests really is what makes, you view the world differently, and you get to work with different people. So, you don't have to just do one route, like you can do a number of different things. And, we have people here who do neuroscience and theater, like we have all sorts of different backgrounds and mixtures. And so you don't have to just pigeonhole yourself into like, I'm a chemist or I'm a neuroscientist. Definitely broaden your interest.
Well, thank you so much, Rachel. Thank you so much. Can we give her a round of applause, please?
Okay.
And now we're going to hand it over to Dr. Jeremy Miller, our next speaker. So same thing with the QR code. You can scan and we will see your questions, and then we can ask Jeremy the questions at the end of the talk. It's all yours, Jeremy.
Great. Thanks for the introduction. I'm happy to talk to you all today about how we are trying to understand cell types here at the Allen Institute. So to begin with, a little bit of history about how cell types have historically been defined. You've heard a little bit about this already. This is an example from the early 1900s about how basically people would look at the brain under a microscope and just draw the cells. So this is from Ramon E. Cajal, who is one of the kind of founders of neuroscience. And he drew this picture from looking under a microscope. There's about 10 or so cells in here. You can see that just by eye, some of them look pretty different. They're in different parts of the section. They have different shapes. Some are big, some are small, and we can characterize cell types that way. We can also see where in the brain they're located if you're going from the frontal cortex versus the temporal cortex. You may have different kinds of cells there. Another way is based on electrophysiology, as you've already heard. You can record the electrical activity in the cell. Here's an example of different kinds of behaviors that different cells may have. Some spike very quickly, some do them consistently, some will do them in bursts, and you can characterize cell types in this way as well. The way that I'll talk the most about is gene expression, where in the past, people would typically look for one bright molecule. So this is an example of a gene VIP, where all of the cells that have this gene are glowing, they show up white, and the ones that don't. And if you choose the right gene for this, you can basically define cells based on the gene that they're expressing. And then there's connectivity. So which cells are touching one another and connect to one another. Certain kinds of cells only want to talk to other cells that are like them. Certain other kinds of cells will only want to talk to cells that are different from them but do a certain thing, and you can characterize cells that way as well. Okay, so how do we characterize cells here at the Allen Institute? What we do is that we measure kind of the same kinds of things that I just mentioned, but using the newer technologies from 2024, recent years. We will define our cell types typically using genes, and then we will characterize the types using the other things like morphology and electrophysiology. Why do we want to use genes to begin with? The first is that they matter during development. So this is just an example that I took. There are many other examples like this of the top left image is showing 3 different areas in a developing mouse brain. The right image is showing the expression of a particular gene across the brain. This EMX2 gene is found the most in the back of the brain. And then the bottom left is showing if you have a mouse that is missing that gene, where do those areas in the brain show up? And you can see that the visual cortex in the back of the brain is much smaller if this particular gene is missing. There are other examples about this, and this is just to say that genes are important. And so if you look at a cell type and you can see certain genes are in that cell type, that can tell you something about what that cell type actually is. Another reason is that there are just a lot of genes. The more features you have of something, the easier it is to characterize it. So this is an example of some apples of different types where we're looking at one color. It's black and white. Pretend like this is one gene. If you then show the same image in color where you have multiple features, imagine this as being multiple genes, it's much easier to tell the yellow ones from the red ones from the green ones. And so we like using gene expression because there are lots of genes that you can use to characterize cells. And then the final reason is that these kinds of gene-based experiments are cheap and fast. You can do one experiment and get thousands of cells with thousands of genes per cell. And so this is just a plot that I took from a blog on single-cell research, where basically each point here represents a different experiment that someone did using these kinds of techniques. The horizontal axis is how many cells were in that experiment, and the vertical axis is how many cell types were found. This is just to say the more cells you measure, the more kinds of cells you can find. And so if we get a lot of genes and a lot of cells, we can kind of find more cell types. Okay, so there are 3 basic approaches using genes for defining and characterizing the cell types. The first is single cell, which is analogous to like a fruit basket. This lets you ask what kinds of cells are they. I'll go into in a minute exactly how we do these experiments, but just to give you the flavor. This one you get lots of data, lots of cells, and we can characterize them, but we lose the context from where they come from. There's another method, spatial transcriptomics, which lets us ask where they're located. So imagine a fruit tart. You actually know, that this is a nice little picture, but imagine this is the brain and not an actual fruit tart. This tells you basically where individual cell types in the brain are located. And then there's a third method called patch-seq, which is, what do they look like and how do they behave? This one, we get one or a small number of cells at a time, but in addition to the gene expression and their location in the tissue, we also get the electrophysiology and the morphology from the same cell, which lets us connect these other features to the gene-based cell types. And we use the combination of these 3 approaches to characterize our cells. Okay, so first, single-cell transcriptomics. Transcriptomics is just a fancy way of saying gene expression. And so, but this method's called single-cell transcriptomics. And this is what we use, as I mentioned, to define cell types at the Allen Institute. The basic approach, so this is a way of measuring basically all of the cells all of the genes in many cells. We start with a brain. We get a section from the brain, like the ones that Caitlin's showing in the back. We can then, from this section, basically take a small piece of that. This section is color-coded by the brain structure. You wouldn't in a real tissue section see that, but it's useful for demonstration purposes here. So then when you get a little section of tissue, it'll look something like this. We basically then just break it down into kind of a bunch of cells get pulled out of there and put into a single test tube, which we then put on a machine, and it tells us for about 50,000 genes and about 10,000 cells, what came from that piece of tissue. We can then repeat that for a bunch of different areas of the brain across multiple individuals to get what we would call like a brain cell atlas of cell types across the human brain. So this is how we get the cells. How do we then define the cell types? This is a toy example showing 6 cells. Obviously, there'll be more than that. But in this case, each circle would represent a cell. Each letter represents a gene. And the size of the letter represents how much of that gene is in that cell. So what we can do is identify cells that have similar gene patterns and call them the same type. So in this case, the 3 yellow cells have more of gene C, the green ones have more of gene A, and the blue one has more of gene B. And so those would represent different cell types. And we can then organize these in what we call a UMAP, where cells with similar gene patterns show up close together in this plot. This does not represent their actual location in the brain, but is a useful way of visualizing the kinds of cell type data that we get. So this is what this plot looks like with 6 cells. When we have 3 million cells across the human brain, it ends up looking more like this. So we've been able to, in a recent study, find about 3,000 cell types organized into these 31 groups in human. We know this is an underestimate. We know we're underpowered in certain areas of the brain from this study. You'll see a different number on the next slide, and that's the reason why those numbers are different. And one thing we've noticed is that cells from the same types usually come, but not always come from the same parts of the brain. So we don't know exactly where these cells come from because there was that step where we took the whole tissue and shoved it into one tube, but we know that many cells of one type will come from like this tissue dissection, and other cells from other types will come from another one in a different part of the brain. We'd like to know exactly where each cell comes from. Unfortunately, with current technologies, human brains are just too big to do this across the whole brain. And so we've also done similar experiments in mouse, where using the same approach, we've found more than 5,000 types of cells in the mouse brain. We think the number of cell types in mouse and human is probably about the same, and just because we're underpowered in our human study, that's why that number is currently lower. But in mouse, in addition to this single cell data, we can do the spatial experiment where we find the exact location of each type of cell. So this is an example tissue section here where each dot is a point, and these actually do represent their specific location in that part of the brain. I'm only showing a subset of the cell types, which is why some of them are gray. But in this case, you find this kind of purple cell type. There's a whole bunch of them kind of clumped up at the top. There's some other ones down here, like these green ones are a similar type. And there's also some down below that are kind of scattered together across different areas of the brain. So some cell types are really clustered, others get kind of scattered around. And by combining these experiments, we can kind of tell that apart. The reason we don't only do this experiment with spatial transcriptomic data is we don't get all of the gene information from this. We only get about 100 to 1,000 genes or so, depending upon the experiment. But by combining these two together, we can actually infer the expression of every gene across every cell, across multiple tissue sections in the brain. And you can too if you want, because we actually provide this freely available online for anyone to look at in what we call the Allen Brain Cell, or ABC Atlas. This is just a screenshot from that tool where we have one example brain with all of the cells color-coded by their type and with lines drawn on that represent the brain regions, like on that one slide I showed before in human, in this case for mouse. Okay, so now we know what the cell types are and where they are, but what do they look like and how do they behave? And this is where patch-seq comes in. So patch-seq is a method that can connect the electrophysiology and morphology to the cell types based on gene expression. The way it basically works is we will get a tissue section from a desired brain region, not quite in the way that I showed before, but for example, in human, you have to use living tissue. You can't use postmortem tissue for this experiment to work. We can then connect an electrode to a single cell body. There are people who actually can see electrodes in single cell bodies in this experiment. I am not one of them, but you can kind of see this little triangle and the little circle, which is actually the electrode connecting onto a particular cell right here. What we then do is we basically put electricity into the cell, simulating a different cell connecting to that and seeing how it behaves. As Rachel mentioned, here's kind of one example of the behavior of one cell. We give it different kinds of electricity for several minutes, and while that's happening, we actually have a dye in the electrode that goes into the cell and fills up the entire cell, not just the cell body, but all the processes that are connecting out into that tissue section. And then when we're done, we do something really cool, which is, I don't actually know how to start this video. Okay. Hold on. Okay, great. Is basically we pull the nucleus, which is the inner core of the cell, out of the tissue, and then drop it into a test tube, and then send that test tube off so we can get the gene expression from the same cell, which we then can use to assign that cell to a specific cell type. And so this is what it looks like kind of for one tissue section. This is one tissue section where 10 different cells were measured in the same piece of tissue using this technique where we have the morphology and the anatomy, the electrophysiology, the gene expression, then mapping those to the different cell types. And we can then color code the cells on this tissue section based on the cell type they were assigned to using this method. And so you can see there's some blue ones that tend to hang out towards the top and some green ones that tend to hang out towards the bottom. And this kind of is a way of tying together all these different kinds of information. Okay, so finally, in addition to these methods for gene-based approaches, there are other methods that we use here that I'm not really going to talk about for characterizing brain cells. One that I find very cool is electron microscopy. This actually lets us look at every molecule in a small piece of brain tissue, where we can, in theory, see every cell and every synapse, assuming we solve the petabyte amounts of data problem to actually do this work. This example here is showing one cell and all of its processes in that tissue section. This then zooms in on one of the sort of processes, and those little bumps are connections of that cell to other cells in this tissue. That is zooming in on one particular connection, and now it goes back to look at the original cell body that connection came from. And so this is a lot more detail that we can get about how all of the cells in a small piece of tissue connect. We don't get the gene expression from this, but we can use patch-seq to help to connect this back to some of the gene level information that I talked about. And then another method is called full neuron morphology, which lets you see the whole cell. Everything I've talked about so far, there's a step where you basically cut a piece of tissue, like in part of the brain. With this method, we can actually look at an entire mouse brain and at a small number of individual cells and see all of the projections kind of across the whole brain. You can imagine that, your legs, most people's legs work. That comes from the brain through the spinal cord at some point. So some of these neurons actually can go pretty far. And this lets you see those. And there are don't worry about what these kinds of cells are, just that the ones on the left and the ones on the right are different kinds of cells. And just by eye, you can see that their patterns look different. Okay, so hold on. So basically using these new technologies, we're learning more about the brain every day. We know the size and shape of the brain, what the different areas of the brain do, how many cells are in the brain, and what it looks like when things go wrong. We don't really know what individual brain cells are doing or how to fix things when they go wrong. And we're trying to learn about the different kinds of cells in the brain, the genes in these cell types, kind of other properties of these cell types, how various kinds of cells connect, and what to call these cell types. And then, the next thing we're trying to work on is how to put all this together. We have all these different kinds of ways we're identifying cells. Some of them link together, but all of them have some amount of incomplete information somewhere. And we want to eventually put this together into kind of a single coherent way that we can describe cell types and that other people, so that we can understand what we mean, and then ideally that you guys can also understand what we mean when we have this final information. So that's all I have. I'll hand it back to Jack for the Q&A.
All right, Jeremy, my first question for you is, what do you think will be the most exciting discovery about the brain coming soon?
The most exciting discovery coming soon? I think that we are just now being able to measure, cells with enough detail that we can start to piece out really subtle things that are going wrong with disease, like in Alzheimer's and other studies. I was anticipating when we first started doing these kinds of single cell studies that there'd be these major things changing in every cell with disease. But it seems like when you look in a brain that is unhealthy, the cells that are still there are pretty healthy, and it's relatively hard to see the differences apart. But now I think we're able to start to see these, and I hope that'll end up translating into some kinds of treatments at some point soon.
Awesome. So my next question is that you talked a lot about morphology or the shape of cells.
Mhm.
So what does that indicate about a neuron, its shape?
I'd say the most obvious, or not most obvious, if a cell has processes somewhere, those are the only places it can connect to. It doesn't always connect to them, but if it's really small, you know it's not connecting to something really far away. If it's pointing things out that are really far away, you know that it probably is connecting to something. So that I would say is one main thing. The shape also can impact like the number of connections and the kinds of things it connects with. So if it's sort of higher up in the tissue, it'll connect to things up there, and if it's lower up, it'll connect to things down there. And since different kinds of cells can be located in different places, the kind of shape of the cell can give you a hint at what other kinds of cells it's talking to.
Perfect. Thank you. So my final question for you, Jeremy, is someone in the audience says that they've heard of brain plasticity. So does this mean that brain cells can change from one type to another?
As far as we know, neurons don't, as far as we know. Some of the glial cells do change states in certain situations. Like if there's something going wrong in the brain, in particular, microglia will change to a different kind of microglia to help sort of whatever's happening there. Neurons, for the most part, don't. They can kind of, the other cells they can connect to will change, but the kind of cell they are will remain the same.
Perfect. Thank you so much, Jeremy. I appreciate it. All right, can we get one more round of applause for our two incredible speakers? So I just want to thank you all again for taking the time out of your day to come here and spend time with us at the Allen Institute. And we all really hope that you walk away having learned something new. But our evening is not over yet. We still have about 30 minutes left. And I'm going to tell you guys a little bit about the activities that we have scattered around in the atrium here. Directly behind you, we have a brain cell station, or brain slides rather, where we have sections of real human and mouse brains, and we will have our wonderful person, Julie, over there to tell you a little bit more about those brain sections. Up here on the mezzanine, just up the stairs there, we have two activities where you can learn more about how we categorize cell types and learn more about the cell types specifically. We have a big wooden puzzle that you can work with. We have a sorting activity with these little Christmas ornaments that light up. It's really cool. You should go check it out. And the food and drinks will be open again, so if you're feeling peckish, you can grab a snack or grab some more drinks upstairs. And again, the bathrooms are just out these doors past the elevators. And if anybody needs to use the elevators to get up to the second floor, you can find somebody with a name badge to escort you up. But yeah, that is all I have. Thank you guys. Feel free to partake in the activities. Thank you. Oh, I'm sorry, I forgot about one more activity. So our speakers, Jeremy and Rachel, will be hanging out at this table here. If any of your questions did not get answered, please come and talk to them. They will be here for the next 30 minutes and are really eager to answer your questions. So thank you again.